In 2017, I was part of an allergen team that discovered a potential biomarker of stress in human saliva. What's more, it seems that the levels of this protein increase actually after people have been subjected to acute stress. The salivary protein that I am studying is called calcium-binding protein spermatid-associated 1, or CABS1 for short. Dr. Bifus, who is an allergen researcher and a professor at the University of Alberta, had done previous work that led us to the study of CABS1. Now, at first the team wondered if the molecule was controlled by the nervous system. But in order to do those kind of studies in humans, we used a psychological stressor. So we subjected participants to psychological stress. For our research, we collaborated with a psychologist in the Southern Methodist University in Texas. He recruited students and subjected them to either acute or immediate stress or prolonged or chronic stress. In each collection of saliva, we asked the students to answer questionnaires about their depression levels, anxiety levels, and how they felt in stress terms. And then these samples of saliva were collected and sent to our university, the University of Alberta, where we studied the levels of CABS1 in those saliva samples. We found that CABS1 levels are relatively consistent over time. However, what's more interesting is that we found that the levels change after the exposure to immediate stress. So, let me elaborate. CABS1 is found at different molecular sizes, and we measured these sizes in kilodaltons. All participants have the presence of a 27 kilodalton band in their samples. When they are exposed to acute stress, the levels of this particular molecule that is 27 kilodaltons increase. Now, what's more interesting is that some people that report to have low levels of stress, they have the presence of smaller than 27 kilodalton bands. So the presence of these bands may indicate resilience to stress. We are very excited about our findings. We think that in the future it may lead to better ways of predicting and diagnosing people with different stress conditions. For example, ongoing studies on CABS1 could lead to the development of a screening tool used by employers to identify stress-resilient employees, let's say air traffic controllers or military personnel. Psychological assessments, questionnaires, could be used alongside this molecular technique to determine who is better suited for particular high stressful jobs.